Okay, I think we can get started now. So in today's webinar, I'll be going over a general overview of Peaks 11, including some highlights in both Peaks Studio and in Peaks Online. So today, this is mainly meant to just, like the title suggests, give a broad overview of everything. And then in following webinars, we will go into more detail about each of the features and different modules of Peaks 11. Okay, so during this webinar, I'll first discuss what's new in Peaks 11 compared to previous Peaks versions. And then I'll give an overview of the different DDA and DIA workflows, followed by some benchmarking data with Xeno swath. And then lastly will be a software demonstration. So the new features in Peaks 11 include our deep learning boosted identification workflows, which I'll show in a few slides, where we've increased identifications by roughly 10% in terms of peptide and protein IDs. We now have streamlined DIA workflows where you can combine spectral library search with DIA DB search, as well as performing de novo sequencing, all within one workflow. We've introduced a DeepNovo based peptidome workflow, which can use for peptidomics or immunopeptidomics to increase the sensitivity and the accuracy in peptide identification. We now have an automated quality control tool that's part of Peaks Online. We have an add-on Glycan Module for glycan profiling and glycoproteomics. We support more instruments, including the new BSI Xeno. And we can now generate spectral libraries to include de novo peptides in addition to database identified peptides. Okay, so we have a completely new graphical user interface for Peaks Studio. So it looks like this. And so at the top now we have tabs which represent each project that is opened. So instead of having all of the projects sort of in this window at the top left, we've now separated them out into different tabs along this top bar here. And then within each project, you can run multiple different types of analyses. And then based on what you've opened up from this window, the different analyses will appear as tabs underneath each of these projects. And I'll go over a few other new features and icons shown in the top a little later in the talk. And the summary page and the other tabs look very similar as previous Peak Studio versions, with just a couple differences. So Peaks Online 11 is accessed through a web browser, but again, you can have multiple different analyses. When you open up a particular project, you can simply create new analyses by checking this plus sign beside the project name. And all of the other features, such as generating spectral libraries or uploading protein sequence databases, can be done in the settings tab at the top. So Peaks Online is a high throughput solution established with all the same Peaks workflows that we have in Studio. One of the main differences is you can have multiple users access Peaks 11 at the same time. It runs on any cluster, multi-CPU machine or cloud server, so you don't have to be on the internet to use it. And this master node can accept multiple computing requests from different users or clients. It could also accept data coming right off the MS instrument using an automated instrument daemon.
It's 202 because wrote the sequence 201.
Okay. In any case, I won't use that one.
Okay, so
I will use
recently, Bernard Kuster's group published this article where they analyzed thousands of proteomes by microflow LC-MS and showed good reproducibility and quantitative analyses with this data. And to highlight the benefit of using our deep learning boosts in our ID workflows, we ran data from this raw data from HeLa, urine plasma, and CSF, and show the percent and increase in IDs for PSMs in blue, peptides in red, and protein groups in green. So in general, we see about a 10% increase in PSMs, between 8 to 10 increase in peptide identifications, and for protein groups we see around 6 to 7% increase in IDs. As I mentioned before, there's a DeepNovo Peptidome workflow that's incorporated into both online and studio, and so this is mainly designed to improve the accuracy and sensitivity of identifying peptides. And so starting from the raw data, we first perform a database search, and this will identify any canonical peptides similar to our PeaksDB search. And so these can be identified using a standard protein sequence database, such as the UniProt database, but it won't identify non-canonical peptides. So we next perform a DeepNovo search, where we do de novo sequencing and then identify any peptides that we consider non-canonical peptides. So these peptides may be generated from splicing events or even expressed from non-coding regions. So regions of the genome where you wouldn't predict an open reading frame. So we categorize these peptides as non-canonical. We use our spider algorithm to find any mutations in these peptides by performing a homology-based search to the database sequences. And we classify these as peptide homologs. And then we also include the peptides identified strictly by de novo sequencing. What's important about this workflow is the deep learning model is trained using peptidomics HLA peptides to improve the accuracy in identification. And also we append the de novo or the DeepNovo peptides to the FASTA file and perform a second round database search in order to have our de novo peptides controlled for by FDR. We then have a final result list of DB, homologs, and DeepNovo peptides, and these can be directly exported for performing binding affinity or immunogenicity predictions in other software. So here's what some of the figures will look like in the DeepNovo Peptidome workflow. So we perform retention time prediction of all peptides and map these as shown here, where we categorize them as database IDs, DeepNovo or homologs, and they're colored accordingly. And we show the regression line for our predicted versus measured retention time for each peptide. We also display the number of peptides and their delta RT from what was expected for the number of database, DeepNovo, and homologs. And by clicking on any particular peptide in the peptide tab, you can see how well the fragment ions match to the predicted values, both in the spectra that has been mirrored with the predicted fragment ions, as well as in the ion match table. We report these values here. A new feature that really improves the confidence of the peptide sequence is we have this CAA percentage filter, and this stands for confident amino acid percentage. So the user can select a certain confident amino acid threshold percentage, And this represents the relative fragment ion intensity required to support each amino acid in that peptide sequence. So in this example, we've set the CAA threshold to 2%. So we require at least a 2% relative fragment ion intensity between each residue in order to get this 100% CAA value. And this is important because in peptidomics and immunopeptidomics, the exact sequence of each peptide is critical in terms of doing downstream analysis, such as binding affinity predictions or immunogenicity predictions. We now have an automated quality control tool that's part of Peaks Online. And so this is an excellent way to assess the quality of the LC-MS data, as well as the peptide protein identifications. And so we can very quickly see whether different attributes either pass or fail in a particular analysis. And this is with respect to a certain acceptance tolerance threshold that is set by the user. And I'll go into this a little bit in more detail in the software demonstration later. We have an option to an add-on feature known as the Glycan Module, where we can perform positional profiling of glycans at particular sites and do glycoproteomics. So shown here is a conserved glycosylation site, an IgG antibody, where these asparagine is heavily glycosylated. And by clicking on the residue, it will pull up this pie chart to show you the relative abundances of the glycopeptides that map to that site. And within the Glycan module, as well as within Peaks 11, we support multiple different instrumentation methods and instrument types. So for glycans, we support Orbitrap, timsTOF, and Orbitrap for HCD, as well as ETHCD. And we also support XenoTOF, CID, and EAD fragmentation types, which has really improved glycoproteomics in terms of getting nice fragmentation ions for both the peptide as well as the glycan molecules. Now included with Peaks 11 is the ability to generate spectral libraries to include the DeNovo peptides as well. So, and this can be done in addition to creating a spectral library with the database identified spectra. So from this drop-down menu, you can select either just a DDA or the DeNovo only result from DDA data. If you select the DDA DB, which is shown here, you can choose whether to include the DeNovo only spectra within the spectral library and set a particular average local confidence threshold, and then choose whether to perform retention time alignment and map to IRT peptides. Okay, next I'll just do a quick overview of the different workflows for DDA and DIA. So here, this is completely new with Peaks 11, where we've incorporated a number of different streamlined workflows for DDA data. So now the user has the ability to strictly do DeNovo or do a Peaks DB search, which can be combined with Peaks PTM and Spider in one workflow. Peaks DB can also, if you have the Peaks Q module, you can perform quantification after database search. And within the DDA workflows, we now include the glycan search as well as DeepNovo Peptidome. Setting up the workflow is fairly similar to the way it was in previous versions of Studio. So you set your error tolerances, enzyme specificities, and PTMs. You set the database and contaminant databases used. We have a peptide length filter. And here is where you would select whether you'd like to enable the deep learning boost for increasing identifications. And then lastly, we have this report page as part of the analysis wizard where you set your different report filters prior to starting the analysis. This is what it looks like in Peaks Online. So fairly similar. So you'd select DDA, the DDA workflows. We have all the same options. And in addition, though, we have this fraction assisted ID transfer quantification. And then again, you set all your parameters here in addition to the report filters. Okay, so last year, our group published a paper in Nature Communications highlighting our streamlined DDA and DIA workflows and also showing how it improves the sensitivity in identifying immunopeptides. So within this workflow, we've integrated this deep learning based solution where we first perform a spectral library search from the raw MS data, and we get our initial peptides identified from the spectral library search. And then any unmatched spectra we can next perform a Peaks database search directly with the DIA spectra. And then any unmatched spectra from both spectral library and database search, we can perform de novo sequencing on. And this is implemented as well on Peaks Online for high performance computing and allowing efficient analysis for high throughput studies. So I'll just go over kind of how to generate a spectral library now in Studio and using it as part of this workflow. And so within the window that shows all the different analyses within the project, you would go to the export tab under the DDA database search. And within that tab, you would select spectral library TSV file and choose whether to include the de novo onlys and map to RT. And then when setting up a search, you load your samples in, select your enzyme and instrumentation types and fragmentation types. And then you would select your workflow. So DIA identification workflow includes, as you can see here, both database and de novo. And then you also have the option to perform quantification with spectral library and database IDs. So the next part of the project wizard would be selecting the spectral library that was generated using the DDA data, and then setting a target protein sequence database for protein inference. All of the spectral libraries that have been uploaded to Studio will appear in this library list, and selecting on any particular library will pull up all of the information. So if you want to perform a direct DIA database search and skip the spectral library, you can, I'll go back. You can unselect this box here, library search parameters, and skip ahead to the database search. Or you can perform both in sequence. So here's part of the project wizard where you set up the database search as part of the DIA workflow. And then lastly, if you like, you can perform a quantification using the IDs from this workflow. And within Peaks Studio 11, we have all the same visualizations as we did in previous versions, such as heat maps and Pearson correlation figures. So now I'll show you some benchmarking data that we've done with Xeno swath data. So SciX published this application note recently highlighting the use of the EvoSep1 system paired with their Xeno swath or Xenotoph instrument for DIA analysis. And so they compared the number of proteins that were identified or quantified from the different sample per day workflows from the EvoSep system. And so here is showing the number of proteins identified, and in the solid part of the bar is the number of proteins that could be quantified across 3 replicates from 200 nanograms of HeLa lysate. And so we took the data so far, we've compared it to the results for the 30 sample per day workflow across 3 HeLa replicates. And so this is performing different aspects of the DIA workflow and comparing the number of identifications. So shown here is doing a direct DIA database search, so skipping the spectral library and just seeing how many proteins we can identify without the use of the spectral library. So this number was just over 4,800 proteins, and close to 4,400 could be quantified across 3 replicates. With spectral library search, we've increased this to over 6,600 proteins, which is comparable to what was identifying using DIA and N as part of this workflow published by SciEx. And over 5,100 of them could be quantified. And when you combine spectral library with the DIA DB search, you can see you can increase this number by roughly 100 identified proteins and significantly more quantifiable proteins. So next, I compared the number of peptide identifications. And so performed the exact same workflow and comparing to the number of peptides identified in this 30 sample per day workflow. And so with direct DIA, we can identify close to 34,000 peptides, whereas in our spectral library search, we can find over 58,000. And then when you combine the two, we're over 64,000 peptides identified, and over 45,000 are quantifiable. And so this is comparable to the numbers reported in the 30 sample per day workflow when using DIA and N. Okay, and lastly, again, just showing how you can increase the number of proteins or peptides identified by performing a database search in sequence with spectral library searching. And so after database searching, we identified, over 90% of the peptides from the spectral library search, but we add additional 163 proteins in the final result after database search. And for peptides, all of the peptides identified in the spectral library will be in the spectral library and DB result, but we've identified initial 7,200 peptides strictly in the direct DIA database search. And then performing de novo sequencing after these 2 workflows, you can further identify 774 peptides from this data set. And another very useful feature within Studio, which is not new to Peaks 11, but it was sort of hidden in previous versions and not many users kind of knew about this, but we have this compare feature, which we've now included an icon to use this feature in the top bar. And you can select any 2 identification results and directly compare them, where it'll generate a Venn diagram and show you whether a protein or peptide was identified in both of these results or just one of them. Okay, so now I'll just switch to the software demonstration and go over a few features in both Peak Studio and Peaks Online. So here's the Peak Studio 11 layout. Here's our Xeno swath data and our Streamlined DIA workflow. So as you can see here, I have the project open with a number of different types of analyses and workflows. So you can perform, spectral library search and direct LFQ analysis, or you can do the entire workflow of performing spectral library followed by DB search and then perform an LFQ. And here's the complete ID workflow where we've done spectral library search followed by DB search, and then the de novo sequencing of any unmatched spectra. So here's what the summary page looks like. We show our FDR curve. We show how well the retention times match to predicted values in terms of our IRT peptides. And we also show a box plot of median delta RT values across all runs compared to our IRT peptides. The protein view looks fairly similar than previous versions. We show the coverage view of different proteins, and new to the peptide tab is the ability to show, I guess, both views. You can show the typical fragment ion spectra and then how well it's matched to the library, or a DB peptide matches to a predicted ion series can be shown in this kind of mirror image. So one of the new things in Peaks 11 is this modify analysis icon. So instead of having to go through the entire setup and project wizard again, you can simply click the modify analysis tool and select which parameters you'd like to change and repeat the analysis. So for example, if you'd like to, instead of performing the library search, you could unselect that and go straight to the database search. All the parameters that were used in the previous analysis are saved. And then you can skip ahead to either de novo or go straight to, you can unselect the de novo and go straight to the report filters. And so the only part of the analysis that has changed will be rerun in the new analysis. So it will, you won't have to redo data refinement and data loading. And if you haven't changed a spectral library search, that won't have to be repeated, and so on. And I'll just show you the compare feature. So clicking on this icon at the top, you can select from different analyses, different results within each analysis. So looking at the Streamlined Xeno swath workflow, we can compare the identifications between the spectral library search with the IDs from spectral library and database search. And then, as you can see, it'll generate a Venn diagram showing you the proteins that are in common from both workflows and those that are unique. These results can also be exported, and we show the protein score distribution here. There are a number of protein and peptide result filters that can also be applied, so you can change the score threshold of proteins that we're comparing, and you can show proteins that are either just common to both results or unique in any particular result. And this is the same for in the peptide tab as well. Sorry, one second. So next, I'll just show you a brief overview of the immunopeptidomics or the DeepNovo Peptidome workflow. So in the summary page, we also have the FDR curve, the statistics table of number of identified proteins, and so on. We could do the precursor mass error calibration. And as I showed before, we have figures showing the how well our peptide retention times match to predicted values. And then in the peptide tab, again, you'll have your list of all peptides are identified in the workflow, but we have this found by column, which will tell you whether it was, say, in that canonical group that were identified in a database search, but we'll also list peptide homologs or DeepNovo peptides in this column as well, and it could be sorted accordingly. And so within, for any particular peptide, you can show the mirror image where you have the predicted fragmentation ions matching to the fragmentation ions from this database identified peptide, as well as the values in the ion match table below. And then also we have the CA percentage column here. So for this particular peptide, if you mouse over it, you can see that we have fragmentation ions for each amino acid in the peptide that are equal to or above that 2% threshold that is set by the user. So in this case, since all of them are supported by fragment ion with relative intensity of 2% or more, this would have a CA percentage of 100. Okay, and here's an example of our Glycan module. So here we've run some IgG samples where we can, and this in particular is with timsTOF data. And so I just wanted to highlight here that we can efficiently identify glycopeptides that map to the 4 different subclasses of these IgG antibodies. And so this is particularly important because although these peptides are isomers and have very similar amino acid sequences and masses, we can differentiate between them across the 4 different IgG subclasses and accurately map glycopeptides to each of the different IgG glycosylation sites. And so, as I mentioned earlier, you can click on the glycosylation site. It will bring up this pie chart showing you the relative percentages of glycopeptide abundances that map to that particular site. And then another tool that can be used is the glycan profiling tool. And so within this tool, if you're interested in any particular glycan, you can select it to be viewed down here. And then you can do glycan site profiling. So for each glycan at this particular site, it will show you the percentage it makes up, or you can switch it to comparing the glycopeptide area values for each glycan that map to that particular site. And then the top scoring PSM for each glycopeptide selected from this table here will be shown on the right. In this case, we only have one sample, so we can't show comparison across replicates. Okay, and lastly, I'll give a brief overview of the quality control tool within Peaks Online. And so here we have a sample filter where within a particular project, you can select which samples you want to include in the QC analysis, as well as selecting any particular attribute to be analyzed. So again, we have attributes related to the performance of the LC-MS, as well as attributes related to MS analysis, such as identification of precursors, peptides, and proteins. So any of these can be selected. And once you save those settings in the summary view, you can quickly assess how many attributes failed in terms of number of samples here. So in this case, for base peak chromatograms, we have 9 samples that have failed. And then down below, we have other statistics tables that will show you number of past attributes. In terms of the DIA DB results, we can show the number of past samples for each of these attributes, and quickly view which samples that this particular attribute has failed in by looking for the red dots in the table. So next, we have a number of different figures that help to assess quality in the data. And the user can set a certain acceptance tolerance. And so this is set as a percentage value and could be with respect to the average value across all samples, or you can set a reference sample if you'd like. And based on which column is selected from this table, you can view the results in figure 1 here. So in this case, selected number of precursors identified across all samples. And as you can see, the number falls within this 20% acceptance tolerance threshold. And so all of these attributes across all samples have passed in terms of number of precursors. If you select base peak chromatogram, you can see the intensities vary quite a bit, and some samples have intensities that fall outside of this 20% acceptance tolerance. And so those values will be in red in the table, indicating that that attribute has failed for that sample. We can also look at TICs across samples, and you can select up to 5 here to compare. And so this allows the user to very quickly assess any major differences in the ion chromatograms across samples, and assess maybe if there was some loading issues or LC problems in certain runs. Lastly, there's the identification QC part within this figure tab. Again, you can select any one of these columns and see how those attributes fall within this acceptance tolerance. So here's showing number of protein groups, number of sequences, or number of peptides, and so on. And then we can generate Venn diagrams showing number of, or the proteins and peptides that were reproducible across samples or replicates, so identified in both. And lastly, we can look at MS1 and MS2 mass error distribution to assess any potential problems in instrument calibration or things like that. And then we have in figure 7, the data completeness, showing you the percentage of replicates with respect to the number of proteins. So in this case, 684 protein groups were identified in 100% of the replicates within this project. And then you have the option to input certain peptides that you might suspect are in the data or should be found across all samples, and just to analyze any differences in the retention time of these peptides across different sample runs. And where there's a gap in the line indicates that the peptide was not identified in that particular sample. To quickly indicate where the peptide may not have been, or what sample the peptide was not identified in, you can click this icon. And if you zoom in, you can see in sample PF or P4 F7, we did not identify this peptide. And then we have the same type of figure to analyze M over Z reproducibility of any standard peptide that's in each of the samples. Okay, so that concludes the software demonstration and the webinar today, and I'm happy to take any questions now.